Dear students, in the last session we have discussed different types of organisms like chrysophytes and dinoflagellates, which comes under kingdom Protista. Kingdom Protista mainly comprises different groups of organisms like chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. We already completed chrysophytes and dinoflagellates. In this session we are going to discuss euglenoids. So euglenoids are the freshwater organisms found in stagnant water. Stagnant water means stored water. The fresh water that is stored. So they usually live in the fresh water that is stored in one particular place. So freshwater organisms found in stagnant water. Instead of the cell wall, they contain the protein rich layer called pellicle, which makes their body flexible. So they do not have the cell wall. Chrysophytes and dinoflagellates contain the cell wall. Here euglenoid does not contain the cell wall. Here the cell is surrounded by the protein rich layer called pellicle. They have the protein rich layer called pellicle, which makes their body flexible. So the body is very flexible due to the absence of the cell wall. And they contain the pellicle. Due to the presence of pellicle and absence of cell wall, their body is flexible and they show metaboly. This is the characteristic feature of the euglenoids. What is metaboly? Movement of euglenoids with the help of flagella by changing the shape is called metaboly. When the euglenoids can move from one place to the other place, they can change their shape. That is the characteristic feature of the euglenoids. So what is metaboly? Movement of euglenoids from one place to the other place by changing the shape is called metaboly. So metaboly is the characteristic feature of the euglenoids. Next, they have two flagella, one is short and the other is long. Like that of the dinoflagellates, euglenoids also contain the two flagella. One flagella is short, another flagella is long. So what is the function of the flagella? Locomotion. Locomotion is nothing but movement. So euglenoids are unicellular organisms. They can move from one place to the other place with the help of flagella. Now coming to the anterior part of the cell contains invagination consisting of three structures, that is cytostome, cytopharynx and reservoir. So the anterior part of the cell contains invaginations consisting of three structures: cytostome, cytopharynx and reservoir. The anterior portion is nothing but upper part of the cell, consists of invaginations. Invaginations are nothing but infoldings. Those infoldings here, this anterior part consists of three structures. Why these three structures are formed in the cell? Because of the invaginations. What are those three structures? Cytostome, cytopharynx and reservoir. Cytostome is nothing but cell mouth. So with the help of cytostome, the cell can take the food material. Cytopharynx, the food can enter into the reservoir through the gullet. Cytopharynx is nothing but gullet and the food is stored in the reservoir within the cell. Next, they contain the photosensitive stigma or eye spot on the membrane of the reservoir. So the membrane of the reservoir consists of photosensitive stigma or eye spot. Next, though they are photosynthetic in the presence of sunlight, they behave as heterotrophs by predating the other small organisms when the sunlight is deprived. Okay, you should remember one thing: euglenoids, they can act as both autotrophs and heterotrophs. What are autotrophs? The organisms which can synthesize their own food material are called autotrophs. Heterotrophs are the organisms, they cannot prepare their own food material but they depend on other organisms for their food, are called heterotrophs. Normally the organism may be either autotroph or heterotroph, but not both. But euglenoids, they have the characteristic feature, that is they can act as both autotrophs and heterotrophs. When the sunlight is there, they can prepare their own food material. When sunlight is deprived, that is in the absence of sunlight, they can act as heterotrophs. How they can act as heterotrophs? By predating the other small organisms. What are predators? Predators are the organisms that can live by killing the other organisms, are called predators. Here they can act as predators. So the organisms which are smaller than euglenoids can be killed by the euglenoids. So though they are photosynthetic, in the presence of sunlight they can prepare their own food material and can behave as autotrophs. When the sunlight is deprived, that is in the absence of sunlight, they behave like heterotrophs by predating the other small organisms. They can eat the other small organisms. They can kill the other small organisms and can eat. Next, the pigments of euglenoids are similar to those present in the higher plants. So, when you are talking about the pigments, you should remember two things regarding the pigments. There are mainly two functions of the pigments. When you take any pigment, the pigment usually gives or impart color to the organism. So, why the different organisms appear in different colors? Because of the presence of different types of pigment. So, pigments impart or the pigment give color to the organisms, and pigment also absorb sunlight. These are the two things that you should remember regarding the pigments. So, plants has the green pigment, it is chlorophyll. So, whatever the pigments present in the higher plants, the same pigments like that of those pigments are also present in the euglenoids. That's why they can prepare their own food material and behave as autotrophs. Next, reproduction is by longitudinal binary fission in palmella stage is found in Euglena. The best example for the euglenoids is Euglena. So, reproduction, how the reproduction takes place in case of euglenoids? By longitudinal binary fission. What is binary fission? Fission is nothing but division. Binary fission means the organism can be divided into two. That is called binary fission. If you take like this, for example, this is the organism. Longitudinal binary fission means here the cell cannot be divided like this, but the cell can divide like this longitudinally, that is vertically. So, longitudinal binary fission. Here the cell divides into two here. So, reproduction is by longitudinal binary fission, and palmella stage is found in Euglena. So, what is palmella stage? Very important. So, palmella stage is usually found in case of euglenoids during reproduction. So, what is palmella stage? So, whenever the conditions become favorable, they can divide by this longitudinal binary fission. So, whenever the conditions become unfavorable, first of all you should understand what are favorable conditions and what are unfavorable conditions. Favorable conditions means, so when the organism can get the food, water, and have the environment, suitable environment for their growth and reproduction. These are called favorable conditions. Unfavorable conditions means lack of food, lack of water, and the environment is not supporting for their growth and reproduction is called unfavorable conditions. So, during unfavorable conditions, this palmella stage is formed. So, what is this palmella stage? So, during unfavorable conditions, the euglenoids first they can throw their flagella. That euglenoids usually contains the two flagella. One is short, another one is long. So, during unfavorable conditions, whenever the conditions become unfavorable, what happens to the euglenoids? So, during unfavorable conditions, the euglenoids thrown their flagella. So, this flagella can be removed from the euglenoids. A number of euglenoids can join together to form the gelatinous mass. So, these are unicellular organisms. Number of euglenoids, they can remove their flagella, and number of euglenoids can join together to form the gelatinous mass. So, once all the euglenoids can join together to form the gelatinous mass, now this gelatinous mass consists of number of euglenoids without flagella. Now, that if you take that gelatinous mass which consists of number of euglenoids, and now each euglenoid can form the double layer cyst. So, number of euglenoids can join together like this. Now, each euglenoid can form the double layer cyst like this. Double layer cyst. Now, each cyst usually consists of daughter euglenoids. Now, each cyst consists of daughter euglenoids. They can undergo binary fission, longitudinal binary fission. So, the gelatinous mass along with cyst is called palmella stage. So, what is palmella stage? During unfavorable conditions, the euglenoids thrown their flagella, number of euglenoids can join together to form the gelatinous mass. Now, in the gelatinous mass, number of euglenoids are joined together. Now, each euglenoid in the gelatinous mass can form the double layer cyst, and which undergoes binary fission. Each euglenoid undergoes binary fission, and each cyst consists of how many daughter cells or daughter euglenoids? 32 daughter euglenoids are present in the each cyst. How 32 daughter forms are formed in the each euglenoid in the gelatinous mass? With the help of a division called longitudinal binary fission. So each cyst consists of 32 forms of euglenoids, daughter forms. Now the gelatinous mass along with cyst is called palmar stage. So the daughter forms consist of flagella again. So this is about euglenoids. Now coming to the slime molds. So we have completed euglenoids. Now coming to the slime molds. So slime molds comes under kingdom Protista. These are the fourth group of organisms which comes under kingdom Protista. Slime molds. Slime molds are the saprophytic protists. What are saprophytes? The organisms which depend on dead and decaying matter are called saprophytes. So parasites depend on other organisms for their food. Saprophytes depend on dead organisms for their food. That is the difference between parasites and saprophytes. So they are saprophytic protists. So they comes under kingdom Protista. So they are saprophytic protists. So the multinucleated mass of protoplasm is surrounded by plasma membrane. Here the slime molds also does not contain the cell wall like that of the euglenoids. So euglenoids and slime mold does not contain the cell wall. Here multinucleated mass of protoplasm is surrounded by plasma membrane. Here what is multinucleated? So here this is the multinucleated mass of protoplasm surrounded by plasma membrane like this. These are called slime molds. So multinucleated mass of protoplasm is surrounded by plasma membrane as they do not have the cell wall. If you take any cell that consists of nucleus and protoplasm, so multinucleated mass of protoplasm is surrounded by plasma membrane as they do not have the cell wall. Next, the body moves or the cell moves along decaying twigs and leaves engulfing the organic material. So here the slime molds, the body of the slime molds or the cell of the slime molds can move along the decaying twigs, that is dry twigs and leaves, engulfing means taking, engulfing the organic material. What is organic material? Carbohydrates, proteins, fats, lipids, these are all present in all the living organisms. If you take the leaves, the twigs are nothing but small stems. So body moves along the decaying twigs and leaves engulfing the organic material. When the body can move along the decaying twigs and leaves, they can engulf the organic material from the twigs and leaves. Engulfing means taking. Organic material is nothing but carbohydrates, proteins, nucleic acids, lipids. These are all organic material which are present in the cells. Whether it be any organism, which may be plants or animals, you can take any organism which contains the organic material. We are going to discuss about this organic material in detail later. Next, under suitable condition, the slime molds form an aggregation called plasmodium. Here what happens? Number of slime molds can aggregate together to form the plasmodium during suitable conditions, which may grow. Here the plasmodium can grow and spread over several feet. So here you should remember, during favorable conditions or during suitable conditions, number of slime molds can aggregate together to form the plasmodium. The plasmodium can grow over several feet and it can spread over the several feet. Next, whenever the conditions become unfavorable, so during favorable conditions, number of slime molds can aggregate together to form plasmodium and the plasmodium may grow and spread over several feet. Next, during unfavorable conditions, the plasmodium differentiates and forms fruiting bodies containing spores at the tip. Now, plasmodium is formed during favorable conditions. During unfavorable condition, the plasmodium, what happens to the plasmodium? It differentiates or breaks down. The plasmodium differentiates and forms the fruiting bodies, and those fruiting bodies contain the spores at the tips. They contain the spores. The spores contain the true cell wall. Normally slime molds does not contain the cell wall, but the spores produced by the slime molds contain the cell wall. The spores contain true walls. Those spores are resistant and survive for many years even under adverse conditions. So why the spores are formed? Please remember, always spores are formed during unfavorable conditions. Why? During unfavorable conditions, no food, no water, no suitable environmental conditions. That's why the spores are formed. The spores can survive without food, without water. Even the condition is too worst also, the spores can survive. That's why during unfavorable conditions, the spores are formed. The spores contain true walls. They are resistant and survive for many years even under adverse conditions. So they cannot be destroyed by climatic conditions. If the food and water is not there, slime mold can die, but the spores cannot die. So they can resistant to adverse conditions. Next, the spores are dispersed by air currents. The plasmodium differentiates and form the fruiting bodies. The fruiting bodies contain the spores or their tips. The spores can be dispersed or released by the air currents. So this is about slime molds. Next, coming to the protozoans.